let's see where we left off. Organization of the brain. The brain is covered with a thick sheath of connective tissue called the meninges. The meninges help protect the brain, keep it anchored within the skull, and resorb the cerebrospinal fluid. They are composed of 3 layers: the dura mater, the arachnoid matter, and the pia mater. Cerebrospinal fluid is the aqueous solution in which the brain and spinal cord rest. It is produced by specialized cells that line the ventricles, or internal cavities, of the brain. Layers of the meninges: skin, periosteum, bone, dura mater, arachnoid matter, pia mater. The human brain can be divided into 3 basic subdivisions: the hindbrain, the midbrain, and the forebrain. Notice that brain structures associated with basic survival are located at the base of the brain, and brain structures with more complex functions are located higher up. The meaningful connection between brain location and functional complexity is no accident. In evolutionary terms, the hindbrain and midbrain were brain structures that developed earlier. Together, they form the brainstem, which is the most primitive region of the brain. The forebrain developed later, including the limbic system, a group of neural structures primarily associated with emotion and memory. Aggression, fear, pleasure, and pain are all related to the limbic system. The most recent evolutionary development of the human brain is the cerebral cortex, which is the outer covering of the cerebral hemispheres. In humans, the cerebral cortex is associated with everything from language processing to problem solving, and from impulse control to long-term planning. Most of the key brain regions described in the following sections are summarized in Table 1.1. Major divisions and principal structures. Forebrain, cerebral cortex. Function, complex perceptual, cognitive, and behavioral processes. Forebrain, basal ganglia. Function, movement. Forebrain, limbic system. Function, emotion and memory. Forebrain, thalamus. Function, sensory relay station. Forebrain, hypothalamus. Function, hunger and thirst. Emotion. Midbrain, inferior and superior colliculi. Function, sensory motor reflexes. Hindbrain, cerebellum. Function, refined motor movements. Hindbrain, medulla oblongata. Function, vital functioning such as breathing or digestion. Hindbrain, reticular formation. Function, arousal and alertness. In prenatal life, the brain develops from the neural tube. At first, the tube is composed of 3 swellings, which correspond to the hindbrain, midbrain, and forebrain. Both the hindbrain and forebrain later divide into 2 swellings, creating 5 total swellings in the mature neural tube. Hindbrain. Located where the brain meets the spinal cord, the hindbrain, or rhombencephalon, controls balance, motor coordination, breathing, digestion, and general arousal processes such as sleeping and waking. In short, the hindbrain manages vital function necessary for survival. During embryonic development, the rhombencephalon divides to form the myencephalon, which becomes the medulla oblongata, and the metencephalon, which becomes the pons and cerebellum. The medulla oblongata is a lower brain structure that is responsible for regulating vital functions such as breathing, heart rate, and blood pressure. The pons lies above the medulla and contains sensory and motor pathways between the cortex and the medulla. At the top of the hindbrain, mushrooming out of the back of the pons, is the cerebellum, a structure that helps maintain posture and balance and coordinates body movements. Damage to the cerebellum causes clumsiness, slurred speech, and loss of balance. Notably, alcohol impairs the functioning of the cerebellum and consequently affects speech and balance. Midbrain. Just above the midbrain, just above the hindbrain, is the midbrain or mesencephalon, which receives sensory and motor information from the rest of the body. The midbrain is associated with involuntary reflex responses triggered by visual or auditory stimuli. There are several prominent nuclei in the midbrain, two of which are collectively called colliculi. The superior colliculus receives visual sensory input, and the inferior colliculus receives sensory information from the auditory system. The inferior colliculus has a role in relative reactions to sudden loud noises. Forebrain. Above the midbrain is the forebrain, or the prosencephalon, which is associated with complex, perceptual, cognitive, and behavioral processes. Among its functions, the forebrain is associated with emotion and memory. It is the forebrain that has the greatest influence on human behavior. Its functions are not absolutely necessary for survival, but are associated instead with the intellectual and emotional capacities most characteristic of humans. During prenatal development, the prosencephalon divides to form the telencephalon, which forms the cerebral cortex, basal ganglia, and limbic system, and the diencephalon, which forms the thalamus, hypothalamus, posterior pituitary gland, and pineal gland. Methods of mapping the brain. Neuropsychology refers to the study of functions and behaviors associated with specific regions of the brain. It is most often applied in research settings, where researchers attempt to associate very specific areas in the brain to behavior, and in clinical settings when patients are treated for brain lesions. Neuropsychology has its own experimental methodology and technology. Studying human patients with brain lesions is one way that researchers have determined the functions of the brain. One problem in studying human brain lesions is that they are rarely isolated to specific brain structures. When several brain structures are damaged, it becomes difficult for researchers to attribute a specific functional impairment to any single brain region. The impairment could just as easily be attributed to any other region that suffered damage. One method for studying the relationship of brain regions and behaviors is to study brain lesions in lab animals. The advantage of this approach is that precisely defined brain lesions can be created in animals by extirpation. Researchers can also produce lesions by inserting tiny electrodes into the brain and then selectively applying intense heat, cold, or electricity to specific brain regions. Such electrodes can be placed with great precision by using stereotactic instruments, which provide high-resolution, 3-coordinate images of the brain. Notwithstanding the ethical or cruelty concerns such studies have raised, they have greatly increased our understanding of comparable neural structures in humans. Another method involves electrically stimulating and recording brain activity. Before operating on the brain, one can stimulate a patient's cortex with a small electrode. This causes individual neurons to fire, thereby activating the behavioral or perceptual processes associated with those neurons. For instance, if the electrode stimulates neurons in the motor cortex, it leads to specific muscle movements. If the electrode stimulates the visual cortex, the patient sees flashes of light that are not really there. By using electrical stimulation, neurosurgeons can thus create cortical maps. This method relies on the assistance of the patient who is awake and alert. Because there are no pain receptors in the brain, only local anesthesia is required. Electrodes have also been used in lab animals to study deeper regions of the brain. Depending on where they are implanted, the electrodes can elicit sleep, sexual arousal, rage, or terror. Once the electrode is turned off, these behaviors cease. Electrodes can also be used to record electrical activity produced by the brain itself. In some studies, individual neurons are recorded by inserting ultrasensitive microelectrons into individual brain cells, recording their electrical activity. Electrical activity generated by larger groups of neurons can be studied using an electroencephalogram or an EEG, which involves placing several electrodes on the scalp. Broad patterns of electrical activity can thus be detected and recorded. Because this procedure is non-invasive, it does not cause any damage, it is commonly used with human subjects. In fact, research on sleep, seizures, and brain lesions relies heavily on EEGs. Another non-invasive mapping procedure is regional cerebral blood flow, or RCBF, which detects broad patterns of neural activity based on increased blood flow to different parts of the brain. RCBF relies on the assumption that when a specific cognitive function activates certain regions of the brain, the blood flow to those regions increases. For example, listening to music may increase blood flow to the right auditory cortex, because that is where music is processed in most individuals' brains. To measure blood flow, the patient inhales a harmless radioactive gas. A special device that can detect radioactivity in the bloodstream can then correlate radioactivity levels with regional cerebral blood flow. This research method uses non-invasive computerized scanning devices. Some of the other common scanning devices and methods of visualization used for brain imaging include CT, computed tomography, in which multiple X-rays are taken at different angles and processed by a computer to cross-sectional slice images of the tissue. PET, positron emission tomography scan, in which a radioactive sugar is injected and absorbed into the body, and its dispersion and uptake throughout the target tissue is imaged. MRI, magnetic resonance imaging, which uses a magnetic field to interact with hydrogen and map out hydrogen-dense regions of the body. fMRI, functional magnetic resonance imaging, which uses the same base techniques as MRI, but specifically measures changes associated with blood flow. This is especially useful for monitoring neural activity, since increased blood flow in regions of the brain is typically coupled with neuronal activation. Parts of the forebrain. The forebrain is the most modern portion of the brain, and in humans forms the largest portion of the brain by weight and volume. The forebrain contains regions derived from the diencephalon, such as the thalamus, hypothalamus, posterior pituitary, and pineal gland. It also includes derivatives of the telencephalon, such as the cerebral cortex, basal ganglia, and limbic system. Thalamus. The thalamus is a structure within the forebrain that serves as an important relay station for incoming sensory information, including all senses except for smell. After receiving incoming sensory impulses, the thalamus sorts and transmits them to appropriate areas of the cerebral cortex. The thalamus is therefore a sensory way station. Hypothalamus. The hypothalamus, divided into the lateral hypothalamus, ventromedial hypothalamus, and anterior hypothalamus, serves homeostatic functions and is key player in emotional experiences during high arousal states, aggressive behavior, and sexual behavior. The hypothalamus also helps control some endocrine functions, as well as the autonomic nervous system. The hypothalamus serves many homeostatic functions, which are self-regulatory processes that maintain a stable balance within the body. Receptors in the hypothalamus regulate metabolism, temperature, and water balance. When any of these functions are out of balance, the hypothalamus detects the problem and signals the body to correct the imbalance. For example, osmoreceptors in the hypothalamus may trigger the release of antidiuretic hormone to increase water reabsorption as part of fluid balance. The hypothalamus is also the primary regulator of the autonomic nervous system and is important in drive behaviors, hunger, thirst, and sexual behavior. The lateral hypothalamus is referred to as the hunger center because it has special receptors thought to detect when the body needs more food or fluids. In other words, the LH triggers eating and drinking. When this part of the hypothalamus is destroyed in lab rats, they refuse to eat and drink and would starve to death if not force-fed through tubes. The ventromedial hypothalamus is identified as the satiety center and provides signals to stop eating. Brain lesions in this area usually lead to obesity. The anterior hypothalamus controls sexual behavior. When the anterior hypothalamus is stimulated, lab animals will mount just about anything, including inanimate objects. In many species, damage to the anterior hypothalamus leads to permanent inhibition of sexual activity. The anterior hypothalamus also regulates sleep and body temperature. Mnemonic. Functions of the hypothalamus, the 4 F's: feeding, fighting, flighting, and sexual functioning. Mnemonic. When the lateral hypothalamus, LH, is destroyed, one lacks hunger, LH. Mnemonic. When the ventromedial hypothalamus, VMH, is destroyed, one is very much hungry, VMH. Mnemonic. When the anterior hypothalamus is destroyed, one is asexual. Other parts of the diencephalon. The diencephalon also differentiates to form the posterior pituitary gland, pineal gland, and connecting pathways to other brain regions. The posterior pituitary is comprised of axonal projections from the hypothalamus and is the site of release for the hypothalamic hormones antidiuretic hormone, ADH, or also called vasopressin, and oxytocin. The pineal gland is a key player in several biological rhythms. Most notably, the pineal gland secretes a hormone called melatonin, which regulates circadian rhythms. The pineal gland receives direct signals from the retina for coordination with sunlight. Basal ganglia. In the middle of the brain are a group of structures known as basal ganglia. The basal ganglia coordinate muscle movement as they receive information from the cortex and relay this information via the extrapyramidal motor system to the brain and spinal cord. The extrapyramidal system gathers information about body position and carries this information to the central nervous system, but does not function directly through motor neurons. Essentially, the basal ganglia help make our movements smooth and our posture steady. Parkinson's disease is one chronic illness associated with destruction of portions of the basal ganglia. It is characterized by jerky movements and uncontrolled resting tremors. The basal ganglia may also play a role in schizophrenia and obsessive-compulsive disorder. Limbic system. The limbic system comprises a group of interconnected structures looping around the central portion of the brain and is primarily associated with emotion and memory. Its primary components include the septal nuclei, amygdala, and hippocampus. The septal nuclei. The septal nuclei contain one of the primary pleasure centers of the brain. Mild stimulation of the septal nuclei is reported to be intensely pleasurable. There is an association between these nuclei and addictive behavior. Amygdala. The amygdala is a structure that plays an important role in defensive and aggressive behaviors, including fear and rage. Researchers base this observation on studies of animals and humans with brain lesions. When the amygdala is damaged, aggression and fear reactions are markedly reduced. Lesions to the amygdala result in docility and hypersexual states. Hippocampus. The hippocampus plays a vital role in learning and memory processes. Specifically, the hippocampus helps consolidate information to form long-term memories and can redistribute remote memories to the cerebral cortex. The hippocampus communicates with other portions of the limbic system through a long projection called the fornix. Researchers originally discovered the connection between memory and the hippocampus through a famous patient named Henry Molaison, known as HM in scientific literature until his death in 2008. Parts of HM's temporal lobes, including the amygdala and hippocampus, were removed in an effort to control epileptic seizures. After surgery, HM's intelligence was largely intact, but he suffered a drastic and irreversible loss of memory for any new information. This kind of memory loss is called anterograde amnesia and is characterized by not being able to establish new long-term memories, whereas memories for events that occurred before brain injury is usually intact. The opposite kind of memory loss, retrograde amnesia, refers to memory loss of events that transpired before brain injury. Go ahead and stop there for tonight.